Hello students, after the completion of the structure of microscope, today we are going to start the cell theory. Cell theory is a very important topic for the 9th class. Students think that it is not important. Actually, cell theory is the basic part of this chapter, so we need to understand it clearly because it can come in 1 mark question, it can come in the 2 marks question also. So if we understand it deeply, we will be able to describe it properly in the question paper. So let's see on the board. Today we are starting the cell theory, and cell theory was given by the German scientist M.J. Schleiden and Theodor Schwann. Actually, Schleiden was the botanist and Theodor Schwann was the zoologist. Botanist means who studies plants and zoologist means who studies animals. Now, see the basic concepts of the cell theory. So in these, the first is that cell is basic unit of structure. That means every structure is made up of cell. Second is the cell is basic unit of function. It is clear to us now that all the functions are going on in the cell, so we can say that cell is the basic unit of function. Third is cell is the unit of heredity. Heredity means to carry character from parents to their children, from parents to the next generation. So the cell is that part of any body, any living thing which carry character from one generation to another generation. Now see, after the Schleiden and Schwann, another scientist gave the cell theory, and this cell theory he given a line called omnis cellula e cellula. The meaning of this line is that every, according to Rudolf Virchow, every cell arises from pre-existing cells. That means not only the specialized cells but all the cells arise from pre-existing cells. That means cell cannot arise from any non-living thing or any dead thing. For the formation of any new cell, there should be one cell. Without that, the new cell cannot be formed. Now we will understand about that how the cell is measured. We measure distance in kilometer, meter, centimeter, millimeter. Similarly, we can measure the size of the cell in the unit called micrometer or microns. Two units are there. Micrometer is represented by the symbol mu m, and the micron is represented by the symbol mu. So these are the very small units, even smaller than millimeter. Let's see how small they are. 1 micron is 1000th part of millimeter, and after the 1000 part, 1 nanometer, another unit is 1 nanometer is 1000th of micrometer. That means it is 1000 part of micrometer. It is very small unit, and in this unit we can measure the size of any cell. Now let's understand about different types of cells. Cells can be of different shapes and sizes. Let's see on the board. First, it is spherical. That means the cell can be rounded or spherical. Second is oval. Cell can be of oval in size, like for example ovum. Ovum means egg. So egg of any living being can be spherical or oval. Second is polygonal. That means it is of different corners. It has different corners, and the liver has this type of cell. Third is cuboidal. That means which will look like a cube. The cell which look like a cube is called cuboidal. Next is spindle shape. This shape is called spindle. So this is the spindle shaped cell, and this type of cell is found in our muscles. Next is cylindrical. Here we can see it is a cylinder. So the cells of xylem are cylindrical. And next is biconcave cell. Biconcave means the cave on both the sides. The cells of RBCs are actually biconcave in size. And third, last type of cells is amoeboid cell. And amoeboid cell can be irregular in shape. And white blood cells. White blood cells of our body are of irregular shape. And red blood cells of our body are biconcave in the shape. Now let's study the difference between plant cell and animal cell. This is very important topic of your 9th class. So we will study it very deeply. Let's start making diagram for both the cells. First, I have drawn the outlinings of both the cells. Normally, all the plant cells are hexagonal in shape, and animal cell we can draw oval or cuboidal, any shape. So let's start. We will start from the nucleus. We will draw nucleus in both the cells. This is the plant cell. This is the animal cell. And what we can see the difference in both the cells, that in animal cell we have made the nucleus in the center of the cell, and in the plant cell we have made the nucleus slightly away from the center. The reason is that in the plant cell there is the presence of some large sized vacuoles which take a large space in the plant cell. So let's first of all we will study about the nucleus. We will draw the outer membrane of the nucleus, and we know that the outer membrane of nucleus is made up of two membranes, outer and inner membrane, like this. Similarly, we will draw in the animal also. We have drawn the outer membrane of the nucleus. So we will mark it. It is called nuclear membrane. And this is the outer and this is the inner membrane. Inside the nucleus we will draw one circular nucleolus. This is nucleolus. Except this, what is the inside liquid is present in the nucleus? Let's draw the liquid, and the name of this liquid or fluid-like substance is called nucleoplasm. Nucleoplasm. This nucleolus have inside it RNA and proteins, and which helps in the formation of ribosomes. So what is the function of nucleolus? The function of nucleolus is to form the ribosomes and RNA synthesis. Now we have seen the complete structure of the nucleus, and what thing is remaining? The most important part is remaining, that is we will draw some thread-like structure in the nucleus, and these thread-like structures are chromosomes. These thread-like structures are chromosomes. So what are the main parts of the nucleus? The main part of the nucleus are, so here we can write like this. This is nucleus. That means nucleus is made up of double membranes, one inner and one outer membrane. Inside it there is a fluid filled, and the name of the fluid is nucleoplasm, and some hereditary material, and this hereditary material is chromosomes, and one small rounded structure is also found in the nucleus, and this rounded structure is called nucleolus, and nucleolus help in the formation of ribosomes and RNA synthesis in the cell. So both the nucleus in both the cells, animal and plant cell, are same, and I think now it is understood by all of you that how to make the diagram of nucleus. Now let's see the outer side parts present in the animal cell and the plant cell. We will start by the endoplasmic reticulum. We know that endoplasmic reticulum part is present outside the nucleus. Like this we will draw it. Endoplasmic reticulum is of two types, some which do not have some rounded molecules on it. These are called ribosomes. These are rounded ball-like structures, and these ball-like structures are called ribosomes. So this endoplasmic reticulum is called rough endoplasmic reticulum. And this one, which do not have any ribosome on it, is called smooth endoplasmic reticulum. And similarly we can draw endoplasmic reticulum here also. So this is rough endoplasmic reticulum, and this one is a smooth endoplasmic reticulum, and these rounded bodies are ribosomes. Now we will draw another structure which is found outside the nucleus and the structure is called Golgi body. This is Golgi body in animals. Golgi body is a very important part of both the cells because it has many functions. Like it helps in the plasma membrane formation, it helps in the lysosome formation, it helps in the formation of enzymes and hormones like proteins, different types of things which are formed by the Golgi body. One more most important thing of Golgi body that it also helps in the acrosome formation in the sperm. The uppermost part of the sperm's head is called acrosome and which is filled by the different types of enzymes which helps in the dissolving the wall of egg. So Golgi body also helps in the acrosome formation of the sperm. Next part is one special part which is only found in the plant cell and it is not found in the animal cell, and this part is called yes, it is very easy because the color of the plant cell is green due to the presence of chloroplast in it. Our next and very important organelle is the mitochondria, and mitochondria are found large in numbers in the animal cell in comparison to the plant cell. So we will draw the mitochondria like outer membrane, then inner membrane which have inner curves, this, and one liquid is also filled in it which is called matrix. In this way we can make the diagram of the mitochondria very easily. First draw the outer membrane and then show curves in the inner membrane like this. This is one curve, this is second curve. Like this, you will make the diagram. So these are the mitochondria which are found both in the plant and the animal cell. Now we will show the liquid filled in the cell. And we know that this liquid is called cytoplasm. This is called cytoplasm. It is easier. Now let's see the difference between the outer membrane of both the cells, plant cell and the animal cell. We have made one very thin membrane surrounding the animal cell, and this thin membrane is called plasma membrane. It is a thin and delicate, flexible outer covering of the animal cell. But the outer covering of the plant cell is different from the animal cell. And it has two layers. One layer, this layer is the plasma membrane, and one outer layer is a thick layer and this thick layer is called cell wall. This is called cell wall is thick and rigid layer. That's why this plant cell cannot change the shape. It is a little bit harder, and it is made up of a substance called cellulose. This is the important question which comes in the exam, that cell wall is made up of cellulose, and it is a rigid outer boundary of the plant cell. And this cell wall is not present in the animal cell. It is absent. And now see, this part I have drawn earlier, and this part is showing vacuole. And vacuole is larger in the plant cell, and in the animal cell vacuoles are smaller, which can be said food vacuoles, which store some food in it. So these are vacuoles in the animal cell. Now another and very important part which is found mostly in the animal cells is lysosomes. Very few are also found in the animal cell, but few in number. Another important part is some rounded structures which are scattered in the cytoplasm in both the kinds of cells, plant and animal cells, and these structures are very, very important structures and are called ribosomes. These are called ribosomes, and ribosomes helps in the process of protein synthesis. Next is some skeletal structures are found, like I have drawn with these blue lines in the diagram, in the animal cell, and these structures are called cytoskeleton. And it is made up of two things. First is microtubules and microfilaments. Next is, and the most important part of the animal cell, which is not found in the plant cell is centrosome. Which is made up of two centrioles. These are called centrioles. And this centrosome helps in the cell division. Helps in the cell division in the animal cell. Now let's understand about the membranes of the cells. Like we have seen here, in the membrane of the cell there is the presence of some pores, and these pores are called plasmodesmata. These are called plasmodesmata. Like this we can draw here in the plant cell. These are called plasmodesmata. Plasmodesmata. These plasmodesmata are the pores which helps in the exchange of material from outside to inside and from inside to outside. These are very important parts. Similarly, the nucleus also has some pores in it, and this is a gap between the nuclear membrane, and this gap is called pore. The name of this pore is nucleopore. And after writing this, now this structure is complete. So this is the difference between plant and the animal cell. Sometimes some animal cells have some villi-like structures. Like this. And these are called microvilli. And these microvilli helps in the attachment of two cells together like this. These microvilli join the two cells together. Some animal cells also have some flagella. And this flagella helps in the movement of the cell. Some animal cells have cilia for the movement and some animal cells have flagella for the movement. So this is the complete structure of both the cells. Now let's discuss some important points in it. See these points. First point. Plant cells do not have any contractile vacuole. Let's understand what is the contractile vacuole. Let's draw contractile vacuole here. This contractile vacuole helps in the removal of extra water from the animal cell. Animal cell has the capacity to shrink itself, and by which this cell removes the extra amount of fluid from the body with the help of contractile vacuole, because this plasma membrane is flexible. But in the plant cell, the wall is rigid, so there is no flexibility in the plant cell, and it cannot remove its extra water with the help of it. Second point is large and permanent vacuole. In the plant cell, vacuoles are large and they are permanent, and in the animal cell, small and temporary vacuoles. Vacuoles are small and they are temporary. And next point is, and the most important point, that centrosome is absent in the plant cell. We have seen that there is no centrosome in the plant cell, and animal cell have centrosome, which helps in the cell division in the animal cell. Now the next is chlorophyll or plastids are present in the plant cell. We have drawn the diagram of the chloroplast here. So chlorophyll is present here, but it is absent in the animal cell. And chloroplast helps in the process of photosynthesis in the plant cell for the formation of food. And due to the presence of the chloroplast or chlorophyll, the color of the plant cell is green. Next point is cell wall present and in this cell wall is absent. We have seen this second layer in the plant cell. It is called cell wall and it is made up of cellulose. It is present in the plant cell but it is absent in the animal cell. Next is it is 100 μm long. Plant cell is 100 μm long and animal cell is smaller than 100 μm. That means plant cell is comparatively larger in comparison to the animal cell. Next is cytoplasm divide by cell plate method. The meaning of this is that when cell divides, when any animal cell divides, there is the formation of cell plate in between. This is called cell plate, and then it forms two cells. And when animal cell divides, it divides by cytoplasm divide by furrowing. Furrowing means the cell constricts on both the sides like this. This is called furrow. This is called furrow, and it makes two cells after dividing with the and the centrioles also helps the division of the animal cell. Next is the plant cell store food in the form of carbohydrate as starch, and the animal cell stores food in the form of glycogen. So this is the most important. It can come as a 1 mark question that plant cell store food in the form of starch, animal cell store food in the form of glycogen. The last point is the lysosomes are less prevalent in the plant cell and lysosomes are more prevalent in the animal cells. And the lysosomes are also called suicidal bags. And these are the structures of plant and animal cell. I hope the difference between plant and animal cell is clear to all of you. If there is any query, you can comment in the comment section. Please like, share and